Hello everyone. This video is about physical characteristics of hormones, hormone latency, post-secretory modifications, and half-life. Along with that, we will be learning some aspects of physiological roles of hormones. Okay, so let's start. First, let's have a quick introduction about hormones. Hormones are essential regulatory molecules produced by various glands and tissues throughout the body. These chemical messengers play a crucial role in controlling and coordinating a wide range of physiological processes. So, hormones are nothing else but chemical messengers. And hormones can be broadly classified into several categories, including peptide hormones. Peptide hormones, these are composed of short chains of amino acids and include insulin, glucagon, and growth hormone. They typically act on the cell surface by binding to receptors and activating intracellular signaling pathways. That's how they work. Then steroid hormones are there. These are derived from cholesterol and include hormones like cortisol, testosterone, and estrogen. Steroid hormones can pass through the cell membrane and bind to receptors within the cell, influencing gene expression. So they have this amazing capability of passing in through the cell membrane and binding into the receptors within the cell, which will eventually lead to the alteration of gene expression. Then amino acid derived hormones are there. These are synthesized from amino acids and include hormones such as thyroid hormones derived from tyrosine and catecholamines like adrenaline and noradrenaline. And the endocrine system comprised of glands like the pituitary, thyroid, adrenal, and pancreas, among others, controls hormone production and release. Hormones regulate various processes, including metabolism, growth, and development, along with that, stress response, immune function, and reproductive function, etc., which we'll be learning later. So, first is about hormone latency, or the physical characteristic of latency of hormones. Latency of hormones. Hormone latency refers to the time it takes for a hormone to exert its full effects on target cells or tissues once it has been released into the bloodstream. So that's how we define latency of hormones. Hormone latency refers to the time it takes for a hormone to exert its full effects on target cells or tissues once it has been released into the bloodstream. Then there are some important points related to this hormone latency, so let's learn about them. Hormone secretion. The latency period begins with the secretion of hormones by endocrine glands or cells in response to specific stimuli. Hormone release can be triggered by factors such as stress, changes in blood glucose levels, or circadian rhythm changes. So all these factors act as some sort of stimuli, which will eventually lead to release of hormones, and this hormone secretion initiates the latency period. So then along with that, transport in blood. Next stage is transport in blood. After secretion, hormones enter the bloodstream. The latency period starts as soon as hormones begin circulating in the blood. The time it takes for hormones to travel from the gland of origin to the target tissue depends on factors such as blood flow and proximity of the gland to the target. Obviously, this hormones, once released from their sources, travel through bloodstream, and the time it takes for hormones to travel from the gland of origin to the target tissue depends on factors such as blood flow and proximity of the gland to the target. And distribution. Hormones are distributed throughout the body via circulatory system. Some hormones may bind to carrier proteins, which can affect their availability to target cells. This distribution process can take minutes to hours. So from now on, we are going to calculate the amount of time it takes. An approximate value, just approximate values of about these hormone latency. So some hormones may bind to carrier proteins, which can affect their availability to target cells. This distribution process can take minutes to hours. So carrier proteins are there. Then receptor binding will be taking place. Once hormones reach their target tissue, they must bind to specific receptors on the surface or inside target cells. We mentioned about steroid hormones. They are capable of entering into the cell. So they will be binding to the receptors present on cytosol, or they will be binding to receptors present on the nucleus, whatever it may be. Some sort of receptor binding will be definitely going to take place. So once hormones reach their target tissues, they must bind to specific receptors on the surface or inside target cells. This binding initiates a cascade of events that ultimately leads to the desired cellular response, which will obviously have some sort of products, or sometimes some inhibitions will be also happening. And once hormones reach their target tissues, they must bind to specific receptors on the surface or inside target cells. These binding initiate a cascade of events that ultimately leads to the desired cellular response. This time, or the time it takes for hormones to encounter and bind to receptors, can vary widely. So don't forget, you are discussing about hormone latency. So these are some important aspects related to this hormone latency. Next, signal transduction. After binding to receptors, hormone trigger intracellular signaling pathways. These pathways involve various enzymes, second messengers, and gene activation depending on the type of hormones. The duration of this signaling cascade can range from seconds to hours. Then gene expression. In the case of steroid hormones such as cortisol or estrogen, the latency period includes the time required for changes in gene expression. Steroid hormones enter target cells, bind to nuclear receptors, and influence the transcription of specific genes. This process can take hours to days. The time duration is highly variable. Then response initiation. The latency period culminates in the initiation of a cellular response. So that is the last stage. This response may involve physiological changes such as altering enzyme activity, cell growth, or neurotransmitter release, whatever it may be, that effect will be there. Then the timing of these responses depends on the complexity of the cellular processes involved. Then some other aspects related to this topic, like duration of effects. The effects of hormone actions can vary in duration. Some hormone responses are transient and short-lived, while others, like those involving growth and development, may have long-lasting effects that persist for days, weeks, or even years. So the time period can be highly variable. There must be some feedback mechanisms. Hormone actions are often subject to feedback mechanisms that regulate their duration and intensity. So, negative feedback loops help maintain hormone levels within a narrow range and prevent excessive or prolonged responses. So, these negative feedback loops are there which prevent excessive or prolonged responses. Okay, so hormone latency encompasses the entire journey of a hormone from secretion to its ultimate impact on target cells. The duration of this latency period can vary greatly depending on the specific hormone, its mode of action, the target tissue, and the overall complexity of the physiological response it triggers. Now we are going to learn some important examples of this hormone latency. Insulin regulation of blood sugar. When you consume a meal containing carbohydrates, your blood sugar level raises. In response, the pancreas releases insulin. We know that. The latency in this process occurs from the time you start eating to when your blood sugar starts to decrease. So that is the time delay or the latency of this insulin hormone. Then insulin facilitates the uptake of glucose by cells, which takes some time to happen. So this is about insulin regulation of blood sugar, which can be stated as an example for this. Then adrenaline in stress response. In a fight or flight response to a stressful situation, your adrenal glands release adrenaline. This hormone rapidly increases your heart rate and redirects blood flow to vital organs. The latency is very short and you feel the effects within seconds. Then growth hormones in growth and development. Growth hormones, which is responsible for growth and development in children, doesn't yield immediate results. We know that. It operates over months and even years, with latency involved in the growth of bones and tissues. So that is going to be an elaborate process. Then stress hormones and the immune response. When you are under stress, the body releases stress hormones like cortisol. These hormones can suppress the immune response. The latency in this case can range from minutes to hours. So immune suppression will be happening as an additional effect of this stress hormones. Now about the next topic, that is post-secretory modifications of hormones, also called post-translational modifications. So post-translational modifications of hormones refers to the chemical modifications that occur to a hormone structure after it is synthesized. Examples of post-translational modifications include glycosylation, proteolytic cleavage, sulfation, and acetylation. So we know that hormones are not synthesized as hormones. They are the end products of a long, journey of synthesis where they start as prohormones and later they undergo some processes which will eventually lead to the creation of hormone. So first stage of this post-secretory modification is the prohormone formation, that is the initial step. So prohormones are inactive precursors that undergo enzymatic modifications, mainly in the endocrine glands, like the secretory glands, to form active hormones. And these modifications typically involve cleavage or addition of functional groups, then transforming prohormones into their biologically active counterparts. The endoplasmic reticulum and Golgi apparatus both play essential roles in the formation and processing of prohormones. The next stage, packaging in secretory vesicles. Once synthesized, prohormones are packaged into secretory vesicles within the endocrine cell. These vesicles also contain molecules called chaperones that protect prohormones from premature conversion into active hormones. Then this also helps to maintain their stability during storage. So you might have heard about the function of protein folding in case of this molecular chaperones. So some sort of similar action is also present in case of hormone synthesis. So these molecular chaperones, they protect prohormones from premature conversion into active hormones. It also helps to maintain their stability during storage. Then cleavage by enzymes. Within the secretory vesicles, prohormones are enzymatically cleaved to the more specific amino acid sequences. This process converts prohormones into biologically active hormones. So convertases, carboxypeptidases, etc. play critical roles in this cleavage process, cleavage by enzymes. Then formation of active hormones. The specific cleavage of prohormones result in the formation of one or more active hormones, the final product. These hormones are smaller and more hydrophilic than their prohormone precursor. The resulting active hormones are released into the bloodstream when the secretory vesicles fuse with the cell membrane during the process of exocytosis. The resulting active hormones are released into the bloodstream when the secretory vesicles fuse with the cell membrane during the process of exocytosis. So that's how they release the contents into outside. Some other aspects: stability and specificity. Removal of unnecessary amino acid sequences and precise cleavage lead to the stability and specificity of hormones. Then hormone storage and release. Secretory vesicles containing active hormones are stored in the endocrine cell until they receive signals such as neural or hormonal stimuli to release their contents. So they remain there after when they receive some sort of signals, they release. How? As I mentioned, they undergo exocytosis. Then temporal regulation. The presence of prohormones and their cleavage into active hormones provide a mechanism for temporal regulation of hormone release. Prohormones can be stored for extended periods and rapidly converted to active hormones when needed. You know that. Then some other important processes involved in this post secretory modifications. Glycosylation is there. That means enzymatic addition of sugar molecules known as glycans to specific amino acid residues within a hormone. Two primary forms of glycosylation is there, like N-linked and O-linked type. Both of these types play pivotal roles in shaping the characteristics of glycosylated hormones. So let's learn about types of glycosylation. N-linked glycosylation. This form attaches glycans in the sense sugar molecules, to asparagine residues, influencing the hormones folding and transport. Then O-linked glycosylation involving attachment to serine or threonine residues. O-linked glycosylation contributes to the structural diversity of hormones. N-linked glycosylation influences the hormones folding and transport, and O-linked glycosylation contributes to the structural diversity of hormones. Then enhanced solubility and stability. These are some important effects of this glycosylation process. Enhanced stability and solubility. Structural modifications will be taking place. Then glycosylation facilitates the proper folding and intracellular transport of hormones within the endoplasmic reticulum and Golgi apparatus. Then glycosylation plays a crucial role in determining the half-life of hormones in circulation. What is half-life? We will be learning later. So glycosylation plays a crucial role in determining the half-life of hormones in circulation. In the presence of glycans can protect hormones from rapid degradation. Some important examples: thyroid hormones, thyroxine T4 and triiodothyronine T3 undergo glycosylation, affecting their transport, binding to carrier proteins, and overall bioavailability. That can be stated as an example. The next process of this post-secretory modification, proteolytic cleavage. This enzymatic process involves the targeted removal of specific peptide sequences from larger precursor molecules, leading to the formation of biologically active hormones. So proteolytic cleavage is instrumental in converting inactive prohormones into their active forms. Cleavage of prohormones often occurs in a tissue-specific manner. Different tissue possesses distinct sets of proteases, allowing for precise regulation of hormone activation based on the tissue-specific needs. Tissue-specific manner action will be there. And in the case of peptide hormones, proteolytic cleavage is essential for their release into the bloodstream. And specific enzymes such as prohormone convertase and carboxypeptidase, we mentioned about these two enzymes earlier, are responsible for the proteolytic cleavage of hormones. There an exemplary case or example. It's the synthesis of insulin. Insulin is initially produced as an inactive precursor called proinsulin. Proteolytic cleavage removes the C peptide from proinsulin, resulting in the formation of mature and biologically active insulin. And why are we mentioning this? Because we are learning about post-secretory modification, and one such post-secretory modification is nothing else but the proteolytic cleavage. So that's why we mentioned about the proteolytic cleavage of this insulin, turning of this inactive precursor into proinsulin, a functional insulin, proinsulin to insulin, which actually takes help of this proteolytic cleavage to remove its C peptide from proinsulin, which will eventually lead to a functional insulin hormone. Then phosphorylation is another method. Another method in the sense, another post-secretory modification happening in case of hormone. And this process involves the addition of phosphate groups. This process involves the addition of phosphate groups to specific amino acid residues within hormone molecules, including influencing their activity, stability, and interactions. Addition of phosphate groups, nothing else. Then phosphorylation is a central mechanism in the transduction of signals. We know that from the extracellular environment to the intracellular domain. It acts as a relay, transmitting information from receptors to the cellular machinery. Upon hormone binding to its receptors on the cell surface, receptor tyrosine kinase often undergoes autophosphorylation. This event triggers a cascade of downstream phosphorylation events, amplifying the signal. So, this phosphorylation is very important in case of transduction of signals, and this acts as a relay. Phosphorylation can activate or deactivate enzymes involved in hormone synthesis or degradation. Phosphorylation can influence the stability of hormone molecule. Phosphorylation of nuclear hormone receptors, such as those for thyroid hormones and steroids, can modulate their transcriptional activity. This influences gene expression and cellular responses. Some sort of transcriptional activity modulation is also happening. Now let's learn about examples. Insulin signaling. Insulin binds to its receptor, initiating receptor autophosphorylation and subsequent phosphorylation of IRS proteins. These phosphorylation events regulate blood glucose levels in concise and orchestrated manner. Next process, acetylation. This process involves the addition of acetyl groups to specific amino acid residues within hormone molecule, influencing their activity, stability, and interactions. So beyond hormone molecules, acetylation extensively impacts histones. And we know histones are, proteins which bind with DNA. So the proteins around which DNA is found. This epigenetic modification regulates gene expression. Which epigenetic modification? Acetylation of histones, nothing else. Acetylation of histones, which can be considered as an epigenetic modification, which regulates gene expression, including those involved in hormone synthesis and response. So acetylation alters the three-dimensional structure of hormones. This modification can influence the charge and hydrophobicity of amino acids, impacting their biological solubility and interaction with receptors. So they influence the three-dimensional structure of hormones, charge and hydrophobicity of amino acids, along with hormonal solubility and interaction with receptors. Then acetylation is regulated by enzymes known as histone acetyltransferase and histone deacetylases. So acetylation can affect the duration and intensity of hormonal signals, providing the fine-tuned mechanism for cellular adaptation. And acetylation plays a role in the regulation of gene transcription and translation. Now let's have an example of this. Acetylation in thyroid hormones. In the realm of thyroid hormones, acetylation plays a crucial role in influencing their interaction with receptors. Ubiquitination. This process involves the attachment of ubiquitin molecule to specific amino acid residues of hormone proteins. Ubiquitination marks hormone proteins for degradation by the proteasome, a cellular structure responsible for breaking down unwanted or damaged proteins. So, ubiquitination acts as a degradation tool, or something which triggers degradation or marks this particular hormone protein for degradation. Then enzymes known as ubiquitin ligase catalyze the attachment of ubiquitin molecules to hormones. And ubiquitination is a mechanism for downregulating hormone receptors. Once ubiquitinated, receptors are targeted for internalization and subsequent degradation. So we have already learned about receptor internalization and receptor desensitization, such sort of mechanisms. Ubiquitination is a mechanism for downregulating hormone receptors. We know there is upregulation and downregulation. Upregulation in the sense more and more receptors will be there, or the affinity of a particular receptor for a ligand will be higher. That can be stated as an upregulation of this receptor. But downregulation is also there. Downregulation in this case works by internalizing that receptor, which is triggered by ubiquitination of hormone. Once ubiquitinated, receptors are targeted for internalization and subsequent degradation. That will eventually lead to degradation. Then ubiquitination contributes to the termination of hormone signaling pathways. Yeah, we mentioned about the internalization of receptors. Then there won't be any binding with receptors. Then there will not be any signaling pathways happening. Then dysregulation of ubiquitination is implicated in various diseases including neurodegenerative disorders and certain types of cancer. Now another example, ubiquitination and growth hormone regulation. In growth hormone signaling, ubiquitination regulates the GH hormone's fate. Upon GH binding, ubiquitin molecules are added to the receptor, marking it for degradation. So dysregulation may contribute to conditions like acromegaly. Gigantism, I guess. Yeah. The next, palmitoylation. This process involves the addition of palmitate, a 16-carbon fatty acid, to specific cysteine residues within hormone molecules, influencing their cellular localization, stability, and signaling dynamics. So, palmitoylation anchors hormones to cell membranes. Palmitoylation influences hormone receptor interaction. Palmitoylation can modulate the stability of hormone molecules. For example, palmitoylation of ghrelin, a hormone regulating appetite, ensures its anchorage to cell membranes. This lipid modification enhances ghrelin's interaction with its receptor, facilitating effective signaling. Remember about this palmitoylation of ghrelin. Next, we are going to learn about the half-life of a hormone. Half-life of a hormone. The half-life of a hormone refers to the time it takes for half of a given hormone to be metabolized or cleared from the bloodstream. Time it takes for half of a given hormone to be metabolized or cleared from the bloodstream. Time it takes for half of a given hormone to be metabolized or cleared from the bloodstream. It is a fundamental characteristic that determines the duration and effectiveness of a hormone's action. Some other definitions are also there. If you find the previous one a bit confusing, learn this one. Hormone half-life refers to the time it takes for the concentration of a hormone in the bloodstream to decrease by 50%. Yeah, this one is a bit more simple. Hormone half-life refers to the time it takes for the concentration of a hormone in the bloodstream to decrease by 50%. It is a critical pharmacokinetic parameter indicative of the hormone's clearance from the body, something related to the hormone's clearance. And it is expressed in units of time. Yeah, obviously it's something which is related to time. Then typically expressed in minutes, hours, or days, depending on the specific hormone. The half-life of a hormone reflects the balance between the rate at which the hormone is produced and secreted into the bloodstream and the rate at which it is metabolized and eliminated from the body. The half-life concept reflects the balance between rate at which the hormone is produced and secreted into the bloodstream and the rate at which it is metabolized and eliminated, or hormone clearance. Factors influencing half-life of hormones. Hormone half-life is a complex parameter influenced by a range of factors including metabolic enzymes, excretion pathways, binding to carrier proteins, feedback mechanisms, receptor interactions, environmental exposure, genetics, circadian rhythms, organ functions, and the aging process. First, we are going to learn about metabolic enzymes, how they affect this half-life of a particular hormone. One of the most influential factors is the presence of activity of metabolic enzymes, primarily located in the liver, that are responsible for breaking down hormones. These enzymes can accelerate or decelerate the metabolism of hormones, directly impacting their half-life. Remember about this metabolic enzymes, which are primarily located in the liver, which create some sort of effects which will break down hormones, which will eventually create an influence on this half-life of a particular hormone. Then excretion pathways. The mode of excretion plays a vital role in determining hormone half-life. Hormones can be eliminated from the body through various routes, including renal, intestinal, or pulmonary. Preliminary in the sense exhalation. So renal excretion in particular can have a significant impact on hormone half-life as the kidneys filtration and excretion rates vary among individuals. You know that. Excretion pathways, how they influence this hormone half-life. Then binding to carrier proteins. Many hormones are there, especially steroids and thyroid hormones, circulate in the bloodstream bound to carrier proteins. These proteins not only transport hormones but also protect them from rapid metabolism. The binding to carrier proteins can extend a hormone's half-life by preventing their immediate clearance. The bound hormone can be gradually released as needed, thus prolonging their effects. Then feedback mechanisms will be there. The presence of feedback mechanisms or feedback loops in the endocrine system can regulate hormone half-life. Negative feedback loops control hormone secretion based on the concentration of hormones in the bloodstream. For example, when blood glucose level rise, insulin is released. Once glucose levels decrease, insulin secretion stops, affecting the hormone's half-life. Then hormone receptors. The presence and density of hormone receptors on target cells can influence hormone half-life. How? Receptor-bound hormones may have a longer half-life as they are protected from degradation. In some cases, internalization and recycling of hormone receptor complexes can prolong the time during which the hormone remains active. Internalization of this, not just receptor, the hormone receptor complex itself is got internalized. So that will prolong the time during which the hormone remains active. Then environmental factors, external factors such as exposure to toxins, pollutants and drugs can affect hormone metabolism and clearance, ultimately impacting half-life. Certain medications, for instance, can induce or inhibit specific metabolic enzymes leading to changes in hormone half-life. Medications and environmental factors also influence the half-life of a particular hormone. Then genetic variations. Genetic factors including genetic polymorphisms in metabolic enzymes and hormone receptors can result in significant individual differences in hormone half-life. Genetic variations can make some individuals more efficient at metabolizing and clearing hormones while others may have prolonged half-lives. Genetic factors, genetic polymorphisms in some metabolic enzymes and hormone receptors can result to significant individual differences in hormone half-life. The next, organ function. The health and function of organs responsible for hormone metabolism and clearance, particularly the liver and kidneys, have a direct impact on hormone half-life. Liver disease or kidney dysfunction can lead to impaired metabolism and clearance, causing hormones to circulate for longer periods. Then circadian rhythms. Many hormones follow circadian rhythms, with their secretion and metabolism varying over a 24-hour cycle. The timing of hormone release and metabolic peaks can affect their half-life. For example, cortisol levels peak in the morning, leading to a shorter half-life during this time. And we know cortisol is something which is related to stress. So stress hormone levels peak in the morning. Next is about some commonly used techniques for determining the half-life of hormones. We will be quickly skimming through these points. Serial blood sampling, a very obvious method, I guess. In this traditional one, blood samples are collected at multiple time points after the administration of a hormone. Hormone concentrations in these samples are measured using assays like ELISA. This data is then used to create a concentration time curve and calculate the hormone's half-life through mathematical analysis. So we will be conducting mathematical analysis to determine the half-life with the help of this concentration time curve, which is actually plotted with the help of taking measurements of the serial blood samples. Multiple time points after the administration of a hormone. We will be administering a particular hormone and then we will be taking blood samples after multiple time points, which will be measured, which will eventually help to create a concentration, which will be used to create a concentration time curve. With the help of mathematical analysis, we could find the half-life of that particular hormone. Next, radioactive tracers and radioimmunoassay. Radioisotopes of hormones are labeled with a radioactive atom and administered to subjects. The decay of a radioactive tracer hormone is monitored over time. Radioimmunoassay technique are then used to quantify the amount of radioactive hormone present in blood samples. The time it takes for half of the radioactive hormone to decay corresponds to the hormone's half-life. Then stable isotope tracers and isotope dilution. Stable non-radioactive isotopes of hormones are introduced into the body, typically in the form of deuterium. Changes in the ratio of labeled to unlabeled hormone in blood or urine samples over time are measured. This data is then used to calculate the hormone's half-life. And examples of hormones and their approximate half-lives. Not accurate values, just approximate values. Insulin half-life around 5 to 6 minutes, a relatively short half-life because of its rapid clearance from the bloodstream. This is essential for precise control of blood glucose levels. An epinephrine or adrenaline half-life approximately 2 to 3 minutes. It's also of a short half-life one. Then cortisol half-life about 60 to 90 minutes, comparatively a longer half-life compared to many other hormones which control processes like metabolism and stress response. Cortisol. And physiological roles of hormone. Some major physiological rules. Functions like that. Physiological roles of hormones. Hormones serve as messengers in the body, orchestrating a wide range of physiological processes. Their roles encompass the regulation of metabolism, growth and development, reproductive function, stress responses, immune system modulation, electrolyte and fluid balance, and the influence of mood and behavior. So let's learn one by one. Regulation of metabolism. One of the central physiological roles of hormone is the regulation of metabolism. Hormones like insulin, produced by the pancreas, play a critical role in controlling blood glucose levels. We know that. So, insulin facilitates the uptake of glucose by cells for energy production and storage. Glucagon, another pancreatic hormone, has the opposite effect, increasing blood glucose levels by promoting the release of stored glucose from the liver. Then, next one, growth and development. Growth hormone produced by the pituitary gland is instrumental in promoting growth and development, especially during childhood and adolescence. It stimulates the growth of bones, muscles, and tissues. Additionally, thyroid hormones such as thyrosin T4 and triiodothyronine T3 play a vital role in regulating growth and development by influencing the development of the nerve system and bone health. Then reproductive function. Hormones are pivotal in regulating the reproductive system. In females, the menstrual cycle is intricately controlled by hormones including estrogen and progesterone. And follicle stimulating hormone and luteinizing hormone produced by the pituitary gland initiate ovulation and maintain pregnancy. In males, testosterone is essential for the development of male sexual characteristics and the regulation of sperm production. And stress response. The body's response to stress is governed by hormones, most notably cortisol. Produced by the adrenal glands, cortisol helps the body respond to stressful situations by increasing energy levels, blood pressure, and reducing inflammation. It is part of the fight or flight response that prepares the body to handle stresses. Then immune system regulation. Hormones play a role in modulating the immune system. For instance, thymosin hormone. Thymosin hormone produced by the thymus gland influences the development and function of T cells, which are key components of the immune response. Then electrolyte and fluid balance. Hormones like aldosterone produced by the adrenal gland help regulate electrolyte balance, particularly the level of sodium and potassium in the body. So aldosterone acts on the kidneys to increase sodium reabsorption and potassium excretion, affecting blood pressure and fluid balance. Mood and behavior regulation. Hormones also influence mood, emotions, and behavior. For example, serotonin, often referred to as a feel good hormone, affects mood and is associated with feelings of well being and happiness. Additionally, the balance of sex hormones like estrogen and testosterone can influence mood and behavior. Then, bone health. Parathyroid hormone and calcitonin are essential hormones for bone health, produced by the parathyroid gland, PTH. Produced by the parathyroid gland regulate calcium and phosphate levels in the body by increasing their release from bones and absorption in the intestine. Calcitonin, produced by thyroid gland, opposes the action of PTH by promoting calcium deposition in bones, thus helping to maintain bone density. Cardiovascular regulation. Hormones influence cardiovascular function, for example, adrenaline or epinephrine and norepinephrine are released by the adrenal glands and play a central role in regulating heart rate and blood pressure. Additionally, atrial natriuretic peptide ANP and brain natriuretic peptide BNP can control blood sugar and blood pressure and fluid balance, not blood sugar, blood pressure and fluid balance, by promoting vasodilation and increasing sodium excretion. This can be stated as a cardiovascular regulation. Then thermoregulation. Hormone also contribute to the regulation of blood temperature, blood and body temperature. So thyroid hormones influence basal metabolic rate, impacting heat production. Additionally, the hypothalamus releases thyrotropin releasing hormone TRH and thyrotropin inhibiting hormone or TSH in response to temperature changes, leading to adjustments in metabolism and body temperature. Then there will be some hormone feedback loops. Many physiological processes are governed by hormonal feedback loops, ensuring a delicate balance between organs and delicate balance within the body. For instance, the hypothalamus pituitary adrenal HPA axis controls the body's response to stress by releasing hormones like corticotropin releasing hormone CRH and adrenocorticotropic hormone ACTH. These hormones trigger the release of cortisol, which then provides negative feedback to inhibit further hormone production once stress is resolved. Then blood cell regulation. Hormones regulate the production and function of blood cells. Erythropoietin, produced by the kidneys, stimulate the production of red blood cells in response to low oxygen levels. Similarly, thrombopoietin influences platelet production, while granulocyte colony stimulating factor regulates the production of white blood cells. Then digestive system control. Several hormones control the digestive system. Gastrin, secreted by the stomach, stimulates the release of gastric acid and promote digestion. Cholecystokinin, released by the small intestine, triggers the release of digestive enzymes from the pancreas and bile from the gallbladder. Insulin and glucagon, produced by the pancreas, regulates blood sugar levels after eating. Then circadian rhythms. Hormones contribute to the regulation of circadian rhythms, the body's internal clock that controls sleep-wake cycles. Melatonin, produced by the pineal gland, increases in the evening to promote sleep, while cortisol levels peak in the morning to wake the body. So that's all about my topic. We mentioned about physical characteristics of hormones like latency, post-secretory modifications, and half-life. What is latency of a hormone? Its definition, some important points related to that, one or more examples, then some important post-secretory modifications and how a prohormone turn into a hormone, then about half-life, what is a hormone's half-life definition, some important points related to this half-life of hormone, along with how to measure this half-life of a particular hormone, then some important examples related to that, along with some important physiological roles of hormones. So that's all about my topic. Thank you for listening.